we will also give some insights as to how basic research transfers to innovative products and processes during this panel. I hope you enjoy our panel. And our first presentation will be by Dr. Arsi Bidani from Lundbeck, Denmark. He will talk to us about how pharmaceutical companies collaborate with the academic sector. The floor is yours, Dr. Bidani.
Thank you so much, Billy. So I will just start by sharing my screen. And please let me know if you can see the slide here. Can you see my slides?
Yes.
In presentation mode now?
Yes, perfect.
So thank you so much, Billy. And I'm really happy to be part of this panel today. And I will talk about how the pharmaceutical companies collaborate with academic sectors. I'm a medical doctor myself, and I had a few years working in the hospital and also a bit more than 10 years now working in the pharma industry. So I hope that, yeah, this presentation will be helpful for most of you. So I wanted to start first by talking about why the academic center or healthcare professionals working in academic centers are really important in drug development. So as you can see here, there are many reasons, and I just wanted to mention the most important ones. So of course, we believe that healthcare professionals have better knowledge of the disease that we would like to treat. They also have the direct contact with patients, leading to a better knowledge of patient needs. I mean, working in a pharma company, we have a limited contact with patient, I would say. Of course, there are some patient insights units that get in touch with patient organizations or sometimes with patients, but it's still limited compared to what healthcare professionals do in their daily work. Third point is like experience with clinical trials. Of course, you all know that clinical trials are running in academic centers and hospitals, and the healthcare professionals are mainly the investigators. So they are really the ones who execute the clinical trials. Another important point is the real world experience. And by that, I mean the efficacy and safety data that they see, not within the studies, but it's more what they see while treating their patients. There could be some differences, and of course, it's always good to hear this feedback. And there are even some trials to assess real-world evidence, and we will come back to that later. And last but not least, the credibility, of course, especially when it comes to communication. Of course, it's always nicer to hear the results of trials from physicians and healthcare professionals, and not from someone from the company, because the healthcare professionals have this credibility toward the scientific community and also toward patients. And I think we all have at least one common goal, which is developing accessible, new, effective, and safe treatment addressing real unmet needs of patients. In this slide, I tried to summarize here the collaboration over the drug development cycle. So the collaboration with academic centers is not really limited to one phase of the drug development, but it goes from preclinical stage to post-marketing. So in preclinical stage, collaboration will be more on selecting the drug candidate and then also generating some preclinical data. In clinical phase, it's more to understand the disease and the target population, meaning what kind of patient population the drug could have the most of benefits, and then also design and execute clinical trials, and then afterward communicate and interpret the results of the clinical trial. Next phase is filling, so it's mostly filing, it's filing, and that's more the interaction with regulatory and payers, and also to convince them about the benefits of this new drug. The collaboration continues also post-marketing, and that's to generate some real-world data, to also have these medical education programs where some top key opinion leaders will educate other members of the scientific community. So as you can see, the interaction is really continuous throughout the drug development cycle. In the coming slides, we will go a bit into the details on how we interact with academia in these different phases. starting by the preclinical phase. So the objective of this phase is mainly to select the drug candidate and then generate some preclinical safety and potential efficacy data, of course. In this phase, actually, the activities are mainly led by the research department in the pharma industry, and the forms of collaboration could be mostly, in the format of research collaboration and preclinical studies. Knowing that not all pharma companies have their own research centers, so sometimes a big part, I would say, of the preclinical studies are done in academic centers. And of course, that's a benefit for both the company and also the academic center, with an experience with a new investigational product and generating new data and new publications. This collaboration becomes even better and even, I would say, wider within the clinical phase. So here we have different objectives, and the main players within the pharma companies are the medical affairs team and also the clinical development team. So as you can see, the first objective is to understand the disease and unmet needs and select the target population. So we generally do some consultation with healthcare professionals and also advisory boards where we gather different experts in the field and discuss some key topics in terms of disease and target population. And this is mainly led by medical affairs, so it's pure medical discussion. The second objective is to design and then execute clinical trials. Again, we do that. We do really listen to the healthcare professionals because they have already some previous experiences with other clinical trials. So we would like to learn from them and avoid a lot of maybe previous mistakes. And we do that through, of course, advisory boards and also through the site selections. Sites for clinical trials are basically hospitals, and the executors are the investigators who are the healthcare professionals. So these kind of activities are led by clinical development. Third objective of this phase is also the communication of the results of these clinical trials, and that's again the medical affairs team. And it could be done through publications where healthcare professionals, physicians and investigators are the lead authors of this publication. It could be under the format of manuscripts or posters. And it could be also through some congress activities like oral presentation, posters, and also symposium. So filing is under the responsibility of regulatory affairs and also market access, where sometimes the pharma companies, they need the support and the endorsement of known experts to convince regulators and payers about the potential benefits of the new drug. So this is also a very good example of collaboration. It could be, we may have this need during the regulatory meetings, so meetings with health authorities, or in some occasion also we do what we call peer advisory boards. The collaboration continues even after the marketing or the commercialization of the new drug, and that's mainly to generate some real-world data. This real-world data could be collected through observational or real-world evidence studies, and that's to confirm what we have seen in the previous clinical trials, because this is more like seeing how the drug is working in the general population, not the selected population in the clinical study. The second objective is to continue the communication about the drug and also publishing the new data we have, again through a standard publication for congress activities. And then the third objective here is the education of target audiences about the disease and the drug, and going back here to the credibility. So it is really a crucial collaboration for pharma companies here to work with known experts to educate the rest of the scientific community about this new drug and the disease, especially when it comes to a rare disease or a disease known with some, I would say, challenges in terms of diagnosis and treatment. So all these activities are also within the medical affairs team. Going back here to the overview, and here you can see the different objectives and the type of collaborations in the different phases. And down also in the slide, you can see the different departments from the pharma company that are involved in this type of collaboration. One last slide to summarize here, and I think that patients need new effective and safe treatment. Healthcare professionals need pharma companies to develop such treatments. And I think also that pharma companies need the knowledge and expertise of healthcare professionals to be able to develop such treatments. So it's really a joint collaboration and joint work to again. We finished with the first sentence, and I mentioned in the first slide also is to be able to develop this accessible, new, safe, and effective treatment addressing real and needs of patients. And I think that's the key word for both academia and also drug development into pharma companies. I hope this was helpful for all of you, and thank you so much for listening to me. And back to you, Pelin.
Dr. Bidani, thank you very much for this wonderful and, in fact, very informative session. I will try to receive the question and answer session questions and answer them at the end of the sessions.
Sure.
So if it is okay with you, thank you very much again, Dr. Bidani. So next we will have Dr. Devrim Demirdora from Akdeniz University Faculty of Medicine, Department of Medical Pharmacology. She will be presenting the living medicines concept, the next generation of human and, in fact, animal healthcare. So Dr. Dora, the floor is yours. Thank you.
Okay, thank you, Dr. Pelin, and thank you I would like to thank the organizing committee for inviting me to this good panel. Now I am sharing. Is it okay for everything? Can you see my slide?
It seems fine. Yes.
Okay. I would like to talk about the living medicine concept under the biotechnological medicines and manufacturing and quality assessments.
Could you please go into presentation mode, because I don't see your
Can you
slide.
see now?
I don't see in presentation mode.
Mm-hmm.
So
is it
I just see
the
the slide
bottom?
very small inside in the screen.
At the bottom right-hand side, you will see a screen, small icon. Maybe you can click that one.
Yeah, it's on the presentation mode.
Yes, please.
Yes, it's now on the presentation mode. Can you see? No, I think
the screen you are sharing is not the right screen. When you share your screen, just maybe change one.
Maybe now. Yes.
No.
Can you see now? No.
No.
When you go to share screen in
yes,
Zoom,
please unshare your screen, Dr. Dora, and reshare it.
Okay.
With the selection of the slides mode, and then from there we can see what we can do.
Okay, I think I am sharing, but now can you see? I will try again. Can you see?
I was informed that if you press the F5 button, Dr. Dora, the F5 button.
Okay. Yes.
No, it's not working.
No, it's not working.
Can we repeat the share mode, Dr. Demirdora? Can we?
Now I am sharing the screen. Yes. Maybe stop share and begin again.
Yeah.
Okay.
Yes.
Yes.
Okay. Share.
Yes. Again.
Okay. Can you see?
We have to be pressing a.
Is it okay now?
Again, it's not on the slide mode.
How can we? It's on the slide mode, but I don't understand the problem. Now I am sharing on my screen. It's saying that you are sharing the screen.
but we have to find the presentation mode.
Yeah, can you put it in presentation mode now?
Yes.
Dr.
Presentation
Demirdora, at the left-hand side, you have 4 icons. I can see the bottom left-hand side.
Yes.
There are 4 icons next to each other. Could you please try pushing the clicking on the last one at the very right-hand side?
Okay.
The right-hand side. Last one.
Slideshow. Yes.
Yes.
Is it?
Yes, it's
No?
on slideshow. Yes. Maybe I can send you the presentation.
No, the problem is it's not
Can
showing
you-
the correct screen. So that's the problem.
Okay. Not stop share and
So
start
you have
again.
to stop sharing.
I am
and
sorry,
then
I am sorry.
share
Yes.
the other screen, I think.
This one is the screen. Yes, share. Is it okay now?
It's the same. The screen
Same.
is your presentation, but we have to have it on presentation mode, I think.
Yes, it's on presentation mode. I don't understand the problem.
Pelin Hoca maybe Devrim Hoca can magnify the slide size.
Okay, I
I mean,
understand.
maybe they can just magnify and then we can continue.
Yes.
Is it now
Hocam,
okay?
Slaytların boyutunu büyütürsek hocam. If we
Can you see now? It's
Yeah,
okay.
it's
Şimdi
wonderful.
oldu
Yes,
mu?
Yes, now
I
it's
think so. Yes.
Oldu mu?
Oldu.
Okay.
Thank you.
Okay, sorry for the problem. Then let's go. In recent years, the knowledge of genetic factors of disease and advances in technology have led to the concept of biotechnological products. And biotechnology simply means the technology of life or the industrial use of life forms. It can be used to make or modify products or processes. Biological systems are used to produce drug substance, or sometimes modern technologies are used to produce drug substance from biological systems. And living medicine is one of the biotechnological drug that consists of a living system that is used to treat a disease. Generally, it consists cells or genetically engineered viruses. There are some different terms for biotechnological products. Biopharmaceuticals are the most widely used terms. Instead of that, sometimes biologicals, biotherapeutics, or biological medicinal products can be used for definition of the biotechnological products. Which products are biological or biotechnological? Recombinant DNA products, therapeutic peptides or proteins, vaccines, hormones, enzymes, cytokines, monoclonal antibodies, immunoglobulins, advanced therapy medicinal products. Under that, gene therapeutics and cellular therapeutics are biological or biotechnological medicinal products. And advanced therapy medicinal products are human medicinal products processed and produced using tissue and gene engineering methods that they are applied to patients to repair damage of the body tissues, to replace the body's normal function, or to ensure the integrity of the disrupted tissue. They are classified under 3 main subjects. One of them is gene therapy medicines. Other one is tissue engineered medicines, and third one is somatic cell therapy medicines. And there is combined ATMPs, which are applied by a medical device. You can see a drug here. It's drug substance inside the drug, and drug product is like a capsule form. Like conventional drugs, biological or biotechnological drugs have drug substance and drug product. Drug substance can be nucleic acids, viral vectors, peptides, proteins, sometimes plasmid DNA, and sometimes cells. They can be complex with a carrier, or they can be complex with delivery systems, or sometimes they have to be formulated for stabilize them. like conventional drugs, they have drug substance, and today, now we will be talking about drug substance. And like for all other medicines, biological medicines must be manufactured following the same quality standards. Compliance with GMP, GLP, and GCP rules are needed. And for biotech manufacturing side, there's some different GMP requirements, special GMP requirements. And in Turkey, all manufacturing facilities for biological products, for drug substance or for drug product side, they can be different from each other and are inspected and licensed by Turkish Medicines and Medical Device Agency Inspection Department. And GMP manufacturing of biologicals needs quality by design-based process development, quality assurance approved documentation, state-of-the-art manufacturing facilities, quality assurance systems to detect deviations and out of specifications, and quality should be designed into the product by the process for biotech products. Here you can see the recombinant protein production scheme. After cloning the suitable or selected gene into the vector, you have to transfect or transform it into the host cell, and you have to make master cell bank and working cell bank. After you identify the quality of your cell bank, you have to make a fermentation process to obtain in large scale your product. And after fermentation, you have to purify them by multi-step processes. And after the purification, you get your bulk product, and you have to be formulated to become a stable product to be given to the patient. For biological or biotechnological products, process is the product. Any change in the vector, in the cell line, in the growth medium, method of expansion, purification methods and conditions can bring a huge variation in the produced product. Because of that, you have to control all of the steps while you are processing your product. As an expression system, you can use different cell lines. Prokaryotic systems are widely used. E. coli is the widely used bacterial system. Yeast, insect cells, mammalian cells, transgenic plants or transgenic animals can be used as expression systems, and they have some difference between each other. Mammalian systems have advantageous agents problems and complex product profiles. Because of that, you have to control mammalian systems instead of raw materials, cell substrates, and downstream purification agents. And because of the complex product profiles, mammalian systems require extensive characterization, and there can be some product-related substances or impurities in the product. Microbial systems have no glycosylation. They are simple systems, and they are good for simple molecules such as interferons or growth factors. Their manufacturing cycle is shorter than the others. They have fewer advantageous agents, and they have less complex product profile. Cell banks are the start point of quality evaluation. It's very important. Characterization of the master cell bank and working cell bank is very important. You have to establish working cell bank from master cell bank and characterize them. Viability, identity, purity, stability, sterility, mycoplasma, advantageous viruses, and retroviruses have to be checked for the characterization. And formulation is one of the very important steps. And the suitability is very important with regards to the stability of the product. Compatibility is very important because of the interaction of formulation with excipients, diluents, and packaging materials is very important. There can be some extractable, leachable impurities from container closure system. Integrity, activity, and strength of the active substance should be demonstrated with the formulation. Preservation of structural integrity and bioactive conformation during and after formulation should be determined. Concentration, pH, temperature, excipients, mixing, shaking, it means that shear force, lyophilization, exposure to air or light, container closure system, storage and route of administration is important for choosing formulations. And stability is also very important. Storage conditions has to be clearly defined because it can be changed from one product to another product. They are all cold change products. They have to be stored under, it changes from product to product, but they have to be stored on like 5 to 8 centigrade degree or minus 20. Drug substance and drug product stability should be carried out using the same container and conditions, which represents the actual storage conditions. And sterilization is very important because you have to sterilize them before giving to the patient, but heat sterilization is not usually applicable. Filter sterilization can be done, but there is some aggregation problem during the filter sterilization. And what are the quality requirements? There is some guidelines for biotech products, also for other conventional products, driven by ICH. ICH Q5, quality of biotechnological products, is given under different chapters, Q5A, B, C, or D. Viral safety evaluation, analysis of the expressing construct stability testing of biotech products, and characterization of cell substrates can be seen on these chapters. And ICH Q6B is the chapter related with specification for test procedures and acceptance criteria for biotech products. There is some tests needed for determination of the quality of the product, with analytical considerations. One of them is physicochemical properties. You have to determine the physicochemical properties of these products. Biological activity is important. Immunochemical properties, purity, impurities, and quantity has to be determined. For these products, for biotech products, extensive state-of-the-art characterization studies should be applied both for drug substance and drug products levels. Due to the complexity and heterogeneity, it is recommended that more than one analytical method applied to each item of the quality to determine and characterize the product. It's needed, more than one test is needed. Statistical analyses are also very important, and appropriate analysis has to be chosen, and validated analytical procedures should be used in the specifications in accordance with the ICH guidelines. Biologics have different physicochemical properties than chemical molecules. They have a small and big complex structure. They have stability problems. They have a lot of modification. They are obtained from the living cells, and it's very difficult to fully characterize the product. But chemical molecules are very easy to handle on. Biological products have a protein structure, which means that they have primary, secondary, tertiary, and quaternary structure. and they have some post-translational modifications, which is related with efficacy and safety of the product. And also tertiary structure is related with the biological activity. And while you are constructing your experiments or constructing your process, it's very important for the protein to become a true translation for the tertiary structure. And biological activity is very important because it's the measure for protein function. There is in vivo and in vitro activity methods. Depending on the biological properties of the product, some different assays can be used, like ligand or receptor binding assays, enzymatic assays, cell-based assays, and functional assays. Biologicals have immunogenicity, and their immunochemical properties have to be determined. There can be some product-related impurities, manufacturing process-related impurities, or post-translational modifications also can cause immunogenicity. The determination of purity depends on an analytical technique, and the results are method-dependent. Process or product-related impurities can be controlled, should be controlled by in-process acceptance criteria or action limits on active ingredients and products. Process-derived impurities are DNA, host cell protein, cell culture components, or antibiotics incited, leaches, and protein A. And product-derived impurities are molecular variants, aggregates, subunits, fragments, oxidized form, deaminated form, and phosphorylated or sulfated forms of the products. Drug substance and drug product specifications should be clearly defined. Appearance and description, identity, purity and impurities, potency and quantity should be defined for specifications. And the setting of specifications for drug substance and drug product is part of an overall control strategy. Which parameters you control of raw materials and excipients in process testing, process evaluation or validation, adherence to GMP, stability testing, testing for consistency of lots. And the aim of the quality assurance is to ensure the quality, safety, and efficiency of the drug product. And finally, there is some challenges with living medicine. I want to sum up all. Classification of the products is difficult. Clinical studies are difficult. How can you target those? Safety and toxicity, how can you identify them? Production site has to be GMP facility. Investment is a big problem because they have complex manufacturing and high cost. Difficulty of logistic strategy, traceability, marketing is different. Donation is a big problem. Product, patient, source, packaging, storage, tewing, and infusion is very problem. And their pharmacovigilance are big problem after given to the patient. And for all the products, safety is first for the patient, and for the safety, high quality products are needed. Thank you for your attention, and thank you for your help in the beginning. And these are the flowers from our campus, Akdeniz University.
Yes, Dr. Demirdora, thank you very much for your presentation. Yes, you have a wonderful, lovely campus. I've been there. Thank you for sharing the pictures also. And it was, in fact, very informative, added on to your perspective on the manufacturing of living medicines, and added on to Dr. Bidani's clinical perspective. We can definitely have a broader sense of how the systems work in this field. Thank you very much.
Thank you.
Next, yes, we have Dr. Zekai Murat Kılıç from University of Manchester, Department of Mechanics, Aerospace, and Civil Engineering. He will talk to us about the importance of manufacturing engineering in tissue regeneration. And I am very excited to hear your speech, Dr. Kılıç. The floor is yours. Thank you.
Okay, thank you very much, Pelin Hocam. Thank you very much for organizing this panel and inviting me to this panel. I'm coming from a manufacturing engineering background, so just from the beginning, I apologize for any kind of terminology mistakes for the medicine-related terms. Yes, so the topic is role of manufacturing engineering in advancing tissue regeneration. So the talk will consist mainly from the studies from the manufacturing group that I'm involved with in Manchester. So let's start. Okay, so the manufacturing engineering is a general. Sorry, I'm just trying to. Yeah, in general view, is designed like to improve the processes in a very, very wide variety of industries, like from aerospace, automotive, and we can count these days. We can count biomedical, construction, energy sectors. So a lot of different industries are making use of manufacturing engineering, and there are like mainstreams of manufacturing engineering. Starting like by machining, the most traditional one, and going into robotics and laser processing, electrical machining, like all the unconventional machining processes, and metal forming or any other material processing. So this manufacturing engineering consists of any material that you can think about in the world, or maybe out of the world even. So the new stream of manufacturing engineering is, of course, additive manufacturing. So of course, you will see why it's important, especially for this talk. And I wanted to start by putting in my view about tissue regeneration. So I'm coming from mechanical engineering, so I like the harder structures. So I choose bone as a point of discussion for this presentation. So this figure shows the how actually the human body is can cope with like loss of bone, even if you help with like a little bit with a scaffold here, with a 3D shaped matching scaffold, and how it can survive itself in a couple of months. In this case, 6 months. And this scaffold does not have any living cells inside, so they just put the polymer material there. So in the tissue regeneration, as I understand, using the living cells is this era. So they want, like we want in manufacturing engineering, to help this tissue regeneration, and the best process that we can think about is the additive manufacturing. The reason is, of course, there are conventional manufacturing techniques which rely on some maybe manual intervened methods, but at the manufacturing, we build layer by layer the structures. So this gives a very, very particular advantage when it comes to any kind of geometry that we need for tissue regeneration. And I put stars in. So there are 7 kind of categories in additive manufacturing which are identified by ASTM, and the stars represent the ones that are very important for the tissue regeneration. So basically, to build biomaterials, and here we see binder jetting. So I will go very fast about what they mean. In binder jetting, there is a powder of materials, and we put kind of glue on top of these materials. We put glue on the region that we want to print. So we just print from bottom to top. And in material jetting, it's a similar sort of concept with binder jetting, but this time we inject material. So you can think about this like an inkjet printing, like on the paper, but in 3D. And powder bed fusion, we put an energy source on a powder, and we kind of melt the powder, and then after it solidifies, it makes a layer. So we build the material like this. And but photopolymerization, what stands for just the tank that this whole thing stays inside. So photopolymerization is like again, there's a layer of material, but we kind of put like a UV light source, if you will, let's say, at the regions that we want to keep for the further printing. So stereolithography, for example, is also under this. So as you can imagine, these 7 processes go like expand to a lot of different machines and processes. And I will focus on material extrusion today, which is the very, very well-known process. And it is very, very diverse, very versatile process. That's why I see it as kind of a feature of like additive manufacturing, helping the tissue regeneration, because you can go up to like as many axes as you can in material extrusion. You don't have any limit, basically, from the space point of view. When I look at the literature, I see a perspective paper about the materials used in line with tissue regeneration. So this paper talks about the bioinks and biomaterial inks. So I think that's kind of a very good description of the materials that are used, because bioinks talk about the soft materials that used for printing the like, let's say, scaffolds or printing the support structures for tissue regeneration, and they have to include cells. So I think that's kind of mandatory. And there are also scaffold-free methods where they only use cells. And today I will talk about biomaterial ink, which consists of kind of a hard structure like scaffolds here, as you can see at the right side. And biomaterial inks are a media for growing cells. So we have to inject the cells on top of these biomaterials after building them. So I will talk about now why these scaffolds are quite important, because of course you can just build one of them using a desktop printer at your home, but I'm pretty sure, almost 100% sure, that you cannot use it for living cells, because of the reasons that I will describe. And that's the importance of manufacturing engineering. So when we look at how we print scaffolds using melt extrusion, that's the scope of this talk. Sorry, I will be faster from now on. So this is a breast implant, and we use CAD model, and then we realize it using this machine. If you have questions in the end, you can just ask me, but there are just typically 3 types of melt extrusion. So we can give any kind of porosity. So that's the takeaway from this slide. We can do any sort of geometry with this kind of system. There are a lot of challenges with melt extrusion. Of course, the speed is one of them. So if you want to go very, very fast speed, that's not a chance. There's no chance because of the vibrations problem, or because of the motors have a limited capacity in the melt extrusion. So you can imagine that it can take up to days to produce even like maybe this size of components using melt extrusion. But maybe with conventional methods, it can take in a couple of minutes to produce the same component. So it's kind of like a trade-off with very complex structure. But you need to be very careful with how you are designing the printing path. Okay, so for the scaffolds, the expectations are several. So one, obviously, non-toxic materials. So previous speakers give a very good view on the stringent requirements of the medical industry. Even much more stringent than aerospace, I understand. And degradation rate, of course. But I want to focus on the mechanical properties here. So when we build some structure, we cannot just build any way we want. We have to choose some parameters. So, and crystallinity of the polymer, if we talk about polymers, basically decides the strength of the structure. And so if you can see here from the top picture to the bottom side, you see a more like a finer resolution image from under the microscope. So this shows actually the structure is more crystalline, and means that it's stronger structure. So we have to be careful in choosing our parameters. So this is one side of engineering, and it involves a lot of testing. Another one is if we want to go into hybrid structures because we want to improve porous structures, we can use, for example, support of another melt extrusion techniques named electrospinning. So this is another way to increase the cell proliferation and to have a better scaffold in a way. And if we want to have better, let's say, reliable techniques, reliable methods in industry, we have to monitor the process. And this is kind of an ongoing research at the moment I'm doing in my group. So we are looking into thermal imaging of the melt extrusion techniques, and as well as optical imaging of the electrospinning techniques. So due to time constraint, I have to pass these slides quite fast, but I'm just trying to focus on the importance of the monitoring of these processes. This is the test bench that we developed my group developed in Manchester. It is plasma assisted. So I think the plasma assisted part is the unique component here, because we build scaffolds layer by layer, obviously, but every layer we have a chance because we have plasma deposition system. So we can deposit plasma layer and then we build another layer. So this gives very, very high improvement, like in the cell proliferation again. And of course, I wanted to talk about all sorts of things about this test bench, but again, due to time requirements, I can't. But the basic idea is we can do a lot of things with the system, including the closed loop monitoring. So this is an ongoing project at the moment. We want to monitor the parameters that are very important for the strength, let's say, as well as the porosity of the structure. So these are the, like at the left side, you can see some ideas about how to improve the closed loop performance, like for example, by using artificial intelligence, digital twins, and of course, by using data fusion. So these can be expanded into many, many different topics, obviously. And as a conclusion, this is very, very futuristic maybe slide, but it has been done in 2016, and people are already trying to look into how to build complex structures which are very important for human body, and one of them is ear, as you can see, and they use like a simple looking scaffold, but of course they also put cells, so the process become in a way hybrid process. And they have a lot of challenges, of course, to control the internal vascularization, for example, and all sorts of other related things. But the important thing here is, like, if we can digitalize this system, and if we can try to keep every effect under control, yes, we can end up with the ideal component in the end. But again, I believe the biggest challenge is how to transfer the laboratory low TRL applications to clinical applications. And I see that this may take even more than like 10, 20 years. So thank you very much for the kind attention. And I want special thanks to my PhD student, Idil, and Fong Nguyen from Bristol, Dr. Fong Nguyen, and Professor Bartolo from Singapore, and of course, Dr. Ferin Kılıç for giving her insights also during preparation. Thank you. I hope I'm not too bad with the time.
Thank you very much. Your timing was perfect. And in fact, it was a very nice perspective from the mechanical engineering side, because you touched base with us on bioink technologies, electrospinning processes, and the stringent requirements that we need to follow by while we are developing our medicinal products. So this also fell in line with Dr. Demir Dora's explanation of the living cells by means of advanced therapy medicinal products, where we use the medical devices as an indispensable part of the product. So in fact, now I'm even more enthusiastic to see how our future projects will follow in the lab together, as we have planned it already. And so I'm very happy, and I'm sure that and you made no mistakes in terminology, and I believe that the ecosystem of multidisciplinary research helps us discover new terminologies for better explaining what we are aiming for in our science. So thank you very much.
Thank you.
And so everybody has spoken. These three speakers have already spoken the best of what the future holds for us in biotechnological healthcare processes and products. So here I will try to explain to you from another perspective. I hope that my screen is good enough. Yes, the multidisciplinary ecosystem reflections on R&D and P&D, because the reason why I'm wanting to explain this to you is that I am carrying two individual personalities in the professional part of my life. One is, as you already know, that I'm following my academic studies and procedures at Ankara University Stem Cell Institute. And the other one is USAK Biotechnology Development and Commerce Incorporated, which I developed by a governmental fund. From there on, I am now also representing the private sector in this field. So, as I have experienced in the several years that I've been going through these two professional lanes that I am following together, the ecosystem is very important in multidisciplinary approaches, because you can have basic research and never be able to carry it through to the large-scale manufacturing. You can never reach it to the clinics. And maybe you do have a groundbreaking innovative idea, and you just cannot formalize it at the end of the day to be able to bring this idea to a real life scenario. So there are, of course, three main pillars. Your ecosystem has to be well established. You have to have a very a fruitful innovation network, and you always have to have strategic alliances where you will have the chance to speak in another language for the same target, for the same goal, to be able to develop one product for the greater good, in fact. So what does this ecosystem look like? Yes, they work all together, but as you can see, the R&D and innovation side has in fact nothing to do with the P&D and the industrialization side. If we do not have integrative parties that are working along with us. So yes, you can be sitting at the academic desk and working at the academic laboratory inside the research institution, but if you do not have the perfect networking, you will never be able to reach the telecommunications and industrial vendors, and you may not be able to engineer your products to bring it to real life. So this is very important. This should be an ecosystem of all parties together to be able to present to governmental bodies and take them as competent authorities for the centralization of the safety, efficacy, and quality of your processes and products, and to be able to serve the society in the best way you possibly can, in fact. So when we look a little bit more into the industry academia collaboration, you can see that it's an everlasting intercircle that continues, that can continue forever if you have built it in the most optimal manner. It seems like a vicious circle, but in fact, it's a ever developing and always living organism. So at one side, you see innovation and knowledge management. At the other side, there is research and experience, which also binds with education, which at the intra and inter-organizational level, if there is a collaborative network, can increase the learning capabilities dramatically, impact. So if you are at one side of the circle and you are just you put all your effort into what you know will be best, you might not have the all of the resources that are necessary to complete your own life cycles, in fact, in research. So, of course, when we're talking about collaborations, there is not only one party that is, you can have a hybrid party of public and private partnerships and collaborations. So I just want to show you what importance there is in public-private partnership models. So the governments, agencies, and the private sectors, the companies can get together to be able to find an easier way out of the financial building and operating burdens of the projects. And you will be able to put your research through to large-scale governmental projects with private funding. And in long decades of times that you can find an environment in which you can share the risk together. Of course, risk sharing will also mean benefit sharing at the end of the day. And in the P3 models, you will see that the most popular one we always come across is the build, operate, transfer model. So a government hands over all operations to a private party for decades and retrieves it all at the end. The similar model is build, operate, and own, the same as the BOT, but the private entity is not required to return the project to the government. This helps to have lengthy, consistent, and ever-developing systems working together, regardless of who is handling the follow-up of the project as it started in the first place. In the types of collaboration, it's not only important to have intra-organizational collaboration, but also inter-organizational collaboration is very important. And what the recent pandemic brought into our lives is the ability to work over the cloud. And of course, partnership collaboration programs are even much more easier and more on a larger scale now, on a global scale with the cloud networking systems. So it is much more easier to collaborate, in fact. So when we look at a joint research program, we see that formal collaborative arrangements aimed at cooperation on pre-competitive R&D projects are subsidized by public funding tools. And if we do not have access to these public funding tools, we might go to contract research where we can directly commercially find firms that are that we can find the support from. And of course, consulting is very important. We might think that we know everything, but that may not be the case, in fact. So something missing there, a third party can see much better and follow you through with all the processes, in fact. So why should we collaborate? Of course, the first utmost important issue is that we need to foster human capital, where the talent pipeline is transferred to the industry, and in return, career opportunities emerge for students who are being, in fact, educated in the first place only to be able to somehow practice their expertise in the real world scenario. so to engineer their information and bring it to the natural scenery, in fact. And there are, of course, some hurdles about financial, operational, reputational, as Dr. Bidani has also expressed, visibility and credibility issues that it could be much more easier to circumvent if you have a hand-in-hand process with collaborative work. And of course, when you're trying to protect your intellectual property, the development of new ideas and somehow applying these ideas will be much easier if you collaborate between industry and academia. So there are, of course, keys to successful collaboration. Motivation is very important. If you cannot motivate one person or anybody who is being educated, you will not have your project on the market at all. This will not reach the general society for serving healthcare. And communication, of course, if you do not communicate what you have done throughout the processes, you will not be able to disseminate your information clearly. So there is, in fact, a clear line between being very transparent and holding on to your know-how, in fact. So you have to really set the lines for these issues. Diversity is also very important. If you have a multifunctional and a multidisciplinary environment, you might be looking from one angle, trying to solve a problem that you just encountered for the first time. And when you see that if there is a multidisciplinary environment, somebody can think outside your box and think inside their box and find the best approach in the fastest way possible, which would be beneficial for each and every one of you, in fact. And of course, as I have said, this causes long-lasting support, in fact. So there is a gap between science and technology, of course, and what everybody is trying to do now in the recent times is they're trying to build a balance between, so that there would be a consistent bridging system. How can we do this? We can design our education programs with a curriculum, with a balanced syllabus that would serve for the industry requirements at the end of the day. So this would start at the undergraduate level, in fact, for me. Consistently transferring knowledge with a skill-based educational program, meaning, yes, they can observe, but they can also put their hands on the processes. Where can they do this? They can do this in your teaching laboratories. They can do this in the research laboratories. They can do this outside of your laboratories with internship circulations, apprenticeships, attending live projects, and this way developing their capacity. I think that the most important point to start collaboration and the notion for collaboration is at the undergraduate level, in fact. So looking at it from the triple helix model perspective, industry, academia, and students have to be somehow connected with each other. With a model that is called entrepreneurial university right now. We have science parks, we have technology transfer offices who will help us out in our technologies, IP settlements, commercialization of our technologies, and meanwhile, we will be able to match the perfect partners from academia and industry while we are raising our students to the highly trained status that they can go in their own disciplines. And of course, as students develop, tomorrow they will be with this industrial exposure that they are in, with their hands-on experiences, they will be tomorrow the academic and industrial people that will be matching together. So this is a notion that is a continuous notion once gained that you can never get away from. And of course, academia is nothing like the industry in some side. Yes, they might be serving the same goals, but in fact, they do differ in many ways. As responsibilities go, I'm sure that everybody will feel for what I'm saying here. There is a big load on the academia side when there are responsibilities. You can barely find time to go into the laboratory, discover what you have already discovered, and try to settle with what you have seen. Maybe you miss out on some of the mistakenly discovered groundbreaking innovations you just because your mind is full. But in the industry, you have applied research and already designed projects that meet the company goals. So it's a more static environment there. And in collaboration in the academia, you are teamwork oriented, you have this freedom. Yes, you have many responsibilities, but you have the freedom to choose with whom you want to collaborate, when and how. And of course, you can discover what you want, and you can be focused on what you want to project and develop impact in the industry. As you can see, it is nothing like that. It's a bit different. But as you can see, the vision, the mission is the same. It's just that they have to get together. And as flexibility goes, as you can see, you can have the freedom to design your own flexibility when you're in the academia. In the industry, it's not always the case. Of course, intellectual freedom goes along with the flexibility. So this hybrid career, this is very interesting for me. I'm trying to develop this in my own company. So varies in this idea varies in the flexibility and supports a variety of different work schedules and offers autonomy to employees to design their own working week in a way that works for both for them and the company policies. I believe that this hybrid career issue can be also reflected on the way a researcher works in the office and in the laboratory setting. So once a researcher has done all the research they can on their table and desktop, they can rush to the laboratory as many days and hours as they like, and come back to research once more and develop their ideas. So I believe that this could be an allegation for another new concept, laboratory office concept that I really enjoy projecting. And the shadow of the valley of death. So research and development. We are in our laboratory researching. We have a wonderful idea that we are very pleased with, and we're doing basic science practices to see if it is working, verify what we have found, try to put onto it as the project follows along. But then we most frequently fall into the valley of death. There, you have to have an industrial partner who holds your hand, puts you through that steep slope up to the peak again, and somehow formulates your basic science ideas to the groundbreaking innovative processes and products. And that's the way it works. So to be able to circumvent the valley of death, I think this is the best model that we can follow. Thank you very much. And I just want to have one word of thanks to the panelists here today. I think this is reflecting the multidisciplinary atmosphere that we are trying to develop in our research or atmospheres. So I really want to thank each and every one of you. And I do really want to thank the participants who took their time to listen to us. I was very happy to have this panel on live show also. And Dr. Elif Kılıç, thank you very much for hosting us and walking us through. This is all I have to say. Thank you very much.
Thank you, Dr. Pelin Kılıç. Thank you all for being here today. I would like to express my sincere gratitude for Dr. Pelin Kılıç for supporting us in every aspect of this Congress. I'm really grateful for your effort. And I owe special thanks for all keynote speakers, moderators, panelists, and speakers audiences. I appreciate that you took time to be here and contribute in the Congress. I hope that you have all found it a fruitful event. Kongremizi bu son panelimizle tamamlıyoruz, kapatıyoruz. Çok kıymetli kongre katılımcıları 2 gün süresince farklı disiplinlerden bildirilerle kongremize katılım sağlayan tüm araştırmacılara çok teşekkür ediyorum. Ayrıca bizi kırmayıp davetimizi kabul eden konuşmacılarımıza, oturum başkanlarımıza ve kongrenin gerçekleşmesinde emeği geçen Kongre Düzenleme Kurulu üyeleri, Kongre Sekreteryası'na çok teşekkür ediyorum. Bize arka planda bizler farklı bilgiler ve tecrübeler edindik açıkçası bu kongrede. Umarım katılımcılarımız için de verimli olmuştur. Yeni çalışmalar ve etkinliklerde buluşmak dileğiyle esenlikler diliyorum. Bye bye.